My name is Krzysztof Mikołajczak. I am from the Institute of Plant Genetics Polish Academy of Sciences in Poznań, Poland. I will succeed in the second call of EPPN 2020 Transnational access, and on this base I gain access to the modern phenotyping facility provided by ALSIA Metapontum Agrobios Research Center located in Metaponto, Basilicata in South Italy.
The current phenotyping facilities in Poland are not sufficient with respect to both throughput and technical level to study roots' reaction to drought during development when the stress is applied in the way that is possible in modern phenotyping facilities. That is why we appreciate that EPPN 2020 activity allows us access to platforms. Such projects additionally give the opportunity to establish international collaboration and exchange knowledge or experience.
My project was aimed to characterize growth and architecture of spring barley roots and their modifications induced by drought. In the preparatory stage, the additional pipe was centrally placed inside the rhizotube, then soil was loaded into tubes with precise weighing. On this way, roots were grown between the rhizotube wall and internal pipe. Altogether, 315 plexiglass rhizotubes were used in the experiment. Each tube was barcoded, ensuring safe identification of each genotype. Water shortage was applied to young seedlings and maintained for 14 days. Imaging started before stress application and was continued during the water shortage and after rewatering. Each plant was imaged sequentially, employing a special near-infrared sensor for root activity and RGB camera. This allowed to evaluate, among others, total root biomass, dynamic of root growth, and root biomass distribution. In the final step, plants were harvested and total green biomass were measured, enabling the calculation of shoot-root ratio.
Recognition of dynamic traits of root system and its potential to grow under water-limited conditions is a grade of importance for a deeper insight into the plant developmental aspects. I believe that genotypes selected in this project by root imaging-based screening can serve as the genetic resources for development of modern, improved barley cultivars in the future. I also intend to correlate obtained outcomes with my already existing dataset that resulted from different omics studies conducted on the same plant material, and altogether it will give a complex knowledge about spring barley response to drought at the whole plant level. I am convinced that we did here in Metaponto a great job, and I'd like to thank my partners from ALSIA research group for help, for support, for kindness, and I strongly believe that we will continue our collaboration. So many thanks guys, and grazie mille ragazzi. Ciao!